Have you ever seen those pesky floaters crossing your vision? Chances are they're proteins, namely collagen, which folds into a triple helix. Wait, how do proteins fold anyway? Proteins are synthesized in unfolded chains of amino acids that are like the beads on a string, and the string then folds into all kinds of shapes, with helices, sheets, barrels, and other mesmerizing structures. Look at this model of insulin. There are actually two chains wrapped around each other, a hallmark of many proteins, such as hemoglobin. Folding is guided by intermolecular forces that push the amino acids into their preferred stable arrangements, called the native shape. This folding pattern defines the function. Compared to regular hemoglobin, fetal hemoglobin has a radically different structure and therefore has a higher oxygen affinity. FYI, you have about 270 million hemoglobins per red blood cell and about 5 million cells per microliter. The human body has over 100,000 different proteins, each with a specific function. Pepsin and chymotrypsin are proteins which digest other proteins. Proper folding leads to proper function. Improper folding, guess what happens? So why should we care? Improper protein folding is the cause of many diseases, including several genetic ones. In sickle cell anemia, a single amino acid substitution from glutamine to valine causes hemoglobin to misfold, which leads to debilitating symptoms. Neurological diseases like Parkinson's and Alzheimer's are also caused by misfolded proteins forming plaques, choking neurons. We could engineer proteins for specific functions, like synthesizing antibodies to stave off deadly viruses. By using Google's AlphaFold, such designs are attainable, and maybe someday it would help us get rid of those pesky floaters. Hey, do you want to fold your own proteins?